today from the Air Force Research Lab at Wright-Patterson Air Force Base in Ohio. And she'll be talking to us about green life science from a DoD lab perspective, biomaterials and biodeterioration. So welcome, Wendy.
Thanks. Good morning. Thanks. Thanks for the opportunity to come and talk to you guys today. It's an honor to be back at my alma mater. I graduated in 2000 with my PhD back when the department was just biology. So I worked in Laura Olson's lab, and my PhD was in peroxisomal transport and protein transport. So I've come very far since then. You'll see that what I do now is entirely different from what I did as a grad student, and that's part of my message today, that you never know where you're going to end up. But you know, if you're well trained, you reach back into that training on a constant basis, and certainly what we do at AFRL is to reach back to basic science on a regular basis too. So I'm going to keep this fairly high level to tell you what we do at AFRL and in my group in particular, but I'll invite questions at the end. So first of all, the Air Force Research Laboratory is about 10,000 employees. We're located in Dayton, Ohio, and there are 8 different technology directorates within AFRL. Most of them are centered at Wright-Patterson Air Force Base. We have a couple out at Kirtland Air Force Base in New Mexico. So you can see we run the gamut of the things we do from, this is the 711th Human Performance Wing, so all things that are related to human physiology and essentially enabling those folks to do their jobs as best as possible. I sit in the Materials and Manufacturing Directorate. There are aerospace systems which deal with fuels and turbines and engines and that kind of thing. We have a whole directorate dedicated to sensors, space and space vehicles, and directed energy, as well as the guys who get to blow things up down in Florida. So, and the Information Directorate. So within the Materials and Manufacturing Directorate, if I can get this to work, we are what we call the one-stop expertise for aerospace materials and systems. And you can see down here that we run the full gamut of different materials. And you can see biomaterials is right here on the end. So within the directorate, we have 12 different research teams, and I am one of the leads for one of those research teams. So we're called Biological Materials and Processing. We have about 30 people on our research team, and we execute about $3 to $5 million a year in research funding. And what we do is to look towards basic science and apply it towards the needs of the Department of Defense. We play in 3 different areas on my team. So you can see here we look at biointerfaces, and that is characterization and predicting the biomaterial-material interactions. One of the reasons why we do that is develop sensor technology. So here's a sensor that is made of carbon nanotubes, but we use a biological recognition element that interacts with that sensor in order to sense different human performance markers like cortisol and neuropeptide Y and that kind of thing. And so understanding how this biology interacts with something abiotic is part of what we do. Second thing that we're involved in is biofilms and microbiomes. So this is characterization, detection, and mitigation of these biomes on aircraft and in fuel systems where they tend to corrode our systems. And of course, that's not a great thing to have to deal with. In addition, we tap into the microorganisms that we bring back from the environment. So we have about 1,500 different organisms in our lab that we've isolated from aircraft and from fuel systems, and we tap into those for new biodiscovery and material synthesis kind of tools. In addition, we have a very strong program in biomaterials and synthetic biology, and this is where I'm going to spend most of my time talking to you about today. So this is moving towards programmable self-assembled materials, materials for what we call cold chain remediation, so that's enzyme ruggedization and in situ synthesis. So if you want to send somebody out overseas with a vaccine or an antibiotic or something, it's really great not to have to refrigerate that thing. And there are many different ways that we try to stabilize enzymes and antibiotics and vaccines in order to make that possible. And the last part here is biophotonic and composite materials. So why do we look at biomaterials? Well, biology, as you all know, is really great at doing a lot of different things. So There are electronic materials, photosystem 2, you know, a really great example of this, something that's very well evolved in order to collect light and transfer energy. We also have photonic materials. Biology is also very good at modifying the optical properties of materials, modifying the spectra coming out of materials, so we're interested in that as well. Then we have materials for materials sake, things like keratin, melanin, which is an electronic material, luciferase, I'm sure you guys are familiar with, and GFP that we can use just because of their unique properties. And finally, biology is really great at structure. So being able to achieve these kinds of hierarchical structures is something that we as synthetic biologists or as biomaterial scientists cannot do just yet. But we look towards all of these things to inspire ways that we can build new materials for the Department of Defense. So we have a strong effort in synthetic biology currently. I'm just going to walk through three kinds of ways that we think about this. First of all is in creating specialty or bulk materials. So basically, you're having an organism spit out whatever you want it to spit out, whether it's a biopolymer or chitin or cellulose or what have you, and being able to make that into a material. So this is high throughput production of monomers, nanoparticles, which can be used for any variety of different types of applications, from decontamination to anti-fouling, creating dopants that can be used for adhesives. I already talked a little bit about biosensors, energetic materials, inks, that kind of thing. Secondly, we think about what we call cellular additive manufacturing. So additive manufacturing in the materials and manufacturing world right now is a big buzzword. We can think about doing this cellularly, so you're taking a biofilm, you're giving it a stimulus and having it produce some kind of material in situ. And this can be used for applications such as self-healing, so you're having the biofilm secrete a polymer in a specific location to heal up a coating that may have been damaged, for instance. You can think about it for anti-fouling if you want to lay down an anti-fouling coating in any particular location. You can think about it for wearable sensors or bandages. And finally, we have here living materials. So this would be a case of cellular responsive additive manufacturing, where you have a biofilm that's secreting the material, and then you give it another stimulus and you can actually get it to actuate. And these are pretty sexy, far out, 20-year-old sci-fi kind of materials, but we think about those things too, because often when you start to think in this space, it inspires what you do back here on the near-term end. So you think about camouflage, and you think more about self-healing and bandages that actually can sense an infection and respond to it immediately and appropriately. Okay, so here's one example that I'll walk you through of biopolymers that we work with. So this is our inspiration here. This is a marine worm called Nereis virens, and it has these incredible jaws that are very strong, very sharp and strong at one end and soft at the other end. And this is a really important property for this organism, because when you think about it, the base of that organism is soft and squishy. This is a big bag of jelly, right? The end of this organism here on these pincers has to be able to pinch a shellfish or something so that it can get food out of that organism. This is the same kind of technology, incidentally, that a samurai sword is built out of too, where the carbon is present at the heel of this sword. It makes it flexible, whereas the tip is very hard. So the worm does it by specifically secreting zinc and hardening this end of the jaw. And so we had seen a paper published on this by Herb Waite's lab in UCSB. We started working with them a little bit. We found out what the protein was that made these worm jaws what they were. It's called NBJP1. And we took that, expressed it recombinantly in E. coli, purified it, and were able to make materials with tunable modulus just by tuning the amount of zinc that we doped into the material. And so basically by controlling the cation and the counterions that you use to dope this material, you can change it from into various conformations and flexibilities. And so what can we do with this? Well, we can think about making materials, for instance, wearable sensors that interface with human skin, where the side that goes against the abiotic side, the actually sensor side, is fairly rigid, whereas the part that goes against your skin is fairly soft. In addition, you can think about staples. These are medical staples that can be biodegradable, or various other types of materials where you would want a modulus and a gradient of hardening across that material. Just wanted to hit up upon this real quick. The Department of Defense has invested very heavily in synthetic biology within the last year, and this will continue for the next 3 years. There was perceived to be a threat from abroad about what folks were doing in synthetic biology, because basically policy abroad isn't what it is domestically. And so the response from the department was that we needed to get smarter about synthetic biology, and we needed to make sure that we had a workforce in the Department of Defense that could understand what was being done abroad in terms of threats for synthetic biology. And so we have a tri-service team, that's Army, Navy, Air Force team of about 100 people, and we're building this competency across the services in synthetic biology. I apologize for the way this looks. It didn't come through from the Mac very well. But at any rate, what we do here is build competencies in these basic research areas, from genetics, genome editing, omics, various characterization tools. We're working with 9 different chassis organisms. So you see, we're not just dealing with E. coli. We do have E. coli in this mix, but we're dealing with different chassis organisms, which we like to think have different superpowers. So each one of these is really good at doing something. And we want to be able to co-opt these for the purposes of either augmenting human performance, building sensors, or building materials. So this is an example of one of the projects that we've got going on in this area. So this is back to that concept of let's have a biofilm build a polymer in situ and have that polymer actuate. And you can see that in order to enable this, you really need to have a bunch of things happen simultaneously. So we're asking questions like, do the biofilms survive and express? Can they make GFP or RFP within a hydrogel? Can we engineer the biopolymer to secrete? So some of these biopolymers are really funky. They'll make inclusion bodies when you try to express them in E. coli or one of those other chassis. So you have to go to great lengths to genetically modify the protein and the secretion pathway in order to get them to secrete. Can we use kind of non-canonical circuits to actuate this material? So can we use light, for instance, instead of your usual IPTG inputs? And finally, how would we actuate the hydrogel? It's hard to modulate ions within the environment in this kind of situation, but how about something like a tyrosinase or some other type of cross-linker that's present there? So I'm just going to wrap up on this slide, and one of the other comments that I wanted to make, especially related to biofilms and microbiomes, is that the degradation of materials is a concern for the Department of Defense, but it's also a concern in the scope of bioremediation. So we're very conscious about the signatures and the trash that we leave behind, the things like hexavalent chrome that's present in our materials, in our coatings, and we want to have good ways to get rid of those things. And often, biology already has good ways to do that, and it's useful to tap into biology, to mine biology to figure out ways to do that. And there are various programs across the Department of Defense, whether it's the Air Force Office of Scientific Research, Office of Naval Research, Army Research Office, Defense Associated Advanced Research Projects Agency, that are involved in funding basic research in order to solve some of these problems. So I would encourage you to keep your eye out on BAAs from the department, where you could use some of your knowledge of plant biology and the really cool things that plants do to apply it to some of these different areas. So with that, I'll stop and I can take questions if there's time.
So questions for Wendy?
I have a question. So when you introduced the biosensors, I thought that was really cool. Could you provide describe some context in which those are used?
In most of those cases, we're either talking about putting them on patients that are being medevaced, so you can have more information coming back to the medical staff on a real-time basis than you would. I mean, you go into a hospital right now and they'll put a pulse ox on your finger, but these are monitoring more types of things than a typical physiological signal like pulse ox. In addition, we work with our special forces guys and our remote pilots. They're in highly stressful situations, and we'd like to know at what point fatigue sets in. If we can monitor that before you actually see fatigue, then you would be able to take these people out of harm's way quicker. Yep.
You mentioned about camouflage.
Yep.
And I'm curious, when just soldiers, say, not Air Force, but when the Army soldiers, say, in the forest, so what's covered on their face is just chlorophyll based material? Because I'm thinking about when you try to make a camouflage for humans versus other animals in the forest, like, often cats and dogs, they do a pretty good job of camouflaging just by using different colors. But for humans, I wonder, do you do anything special?
I would say that it's not a special material necessarily, but the patterns are what we learn from the environment. So, for instance, people have studied the cryptic coloration in cephalopods for a really long time to determine when they sit down on a substrate how their skin models. They actually only have about 16 patches of skin on their backs, and they're able to modulate each one of those patches specifically. And so, how do you translate that to camouflage for a person wearing a suit of some sort? Not that we're even there, and then how do you, again, backtrack that to more near term where you're designing new uniforms for people such that they're going into a new environment. How does that modeled pattern have to match where they're headed? So you see guys in the airport, for instance, headed off to the desert. They'll be wearing much different uniforms than if they're headed out to sea or headed out to, the rainforest, for instance. So.
All right. Thank you, Wendy.
Okay.